Good afternoon, girls and boys. Can you hear me? 下午好。李英杰下午好。李一彤下午好。黎明下午好。叶微玉下午好。陈丽萍下午好。小七下午好。赵英儿下午好。花妹妹下午好。张富明下午好。华娟飞下午好。黄小恩下午好。耶、秀梅下午好。新梅下午好。莎莎下午好。We will begin our class. Okay. Yeah, can you see my PPT?
Yes, 老师。
Yes, 老师
Yes, 老
Okay.
师。
This class that we major study the two experiment, one experiment for the blood group identification. Yeah. Can you hear me?
Yes, 老
What's
师。
that? Yeah. Yeah. I'm sorry, my internet some wrong. Yeah, now can you see my PT? Yeah.
Yes, no, sir.
Okay. The first in this class we will study the first experiment that the blood group identification and also some of the blood calculation factors. They are which that we study that the blood group identification. This experiment that first that the purpose that master method of the ABO blood group identification and also that understand the series of the consequences caused by the blood type incompletely during the blood transfusion. So we know that we have studied that the chapter 3, the blood we have known that they are doing the blood transfusion should keep the blood group in blood group compatibility. Yeah. First that we review the knowledge of the blood group. The blood group we have known that nowadays also that in many kind of the blood group, and there we majorly that study that the ABO blood group and the RH blood group, and there normally that we know that use the blood group majorly that ABO blood group, and then there are this class. I hope you know that how to do. How to identify ABO blood group, and this the experiment, the nature of the experiment. First, that we have known that in the red blood cell also that we have known that red blood cell on the red blood cell membrane there have many some the antigens, and also there antigens there we can see here this majorly that the antigen A antigen, and also there we can see there is also that the we have known that we can see that this like that this person that blood is the blood red blood cell is antigen is A antigen, and there are also that we have known that also that in the plasma there is also some the antibody we can see here that like that this anti-B antibody we can see here that when the A antigen and the anti-B antibody together, we found that they cannot ovulation because there we can see here that this is also cannot bound the antibody cannot bound in the antigens, and also they are cannot antigens, and also they are this not can ovulation. And also then we can see here that if this antigen A, and also there this then the plasma antibody is also anti-A antibody, and then anti-A antibody and then can bind in the A antigens, and also that this we can see this antibody can bind in one antigens on the red blood cell, on another red blood cell. We can see here that this collect they need to that more red blood cell ovulation. Yeah. So we according to this nature of the identify the red blood cell that is the same type of the antigens and the antibodies then can ovulation. Yeah. And then this different type that like that only that A antigen and can binding anti-A antibody. And also that A antigen cannot bind in the different type that the anti-B antibody cannot bind in. So that there we have known that there is then according to that we can see that if we know that if we should task and identify the people's red blood cell which types of the A antibody or A antigens or B antigens, then we can base this base reactions, and also then we can if there are have the anti-B and anti-A antibodies, and then we can by this reactions we know that the type of the antigens of the red blood cell membrane. Yeah, can you understand? Yeah. So that we know that this is the nature of the identify the red blood ABO blood group. Yeah. And there, before we started the red blood, this experiment, with knowledge that the red blood cell, the red blood cell blood group, and then we measured that ABO blood group, and also some of the RH blood group, and measured that the ABO blood group. I hope you know that according to the the absence of or the presence or absence of the A and the B antigens on the red blood cell membrane, and they can be classified the full type of the ABO blood group. There will be only A antigens on the red blood cell membrane, and there this blood is a type A, and there is only B antigens. They are type B. And there with the both A and both B antigens in the red blood on the red blood cell membrane, this blood is type AB, and both A and both B antigens were not on the red blood cells. There is a type O blood group. So here that according to the antigens, A antigens and B antigens on the red blood cell, the ABO blood group is 4 types: A type, B type, and AB type, and O type. Yeah. This remember? Do you remember this?
Yes, 老师. Yes.
This is very important. Also, that in the clinic, if you also in the blood group, yeah. And also that we have know that the ABO blood group, and also that the natural antibodies. It means that like that, if the people in the red blood cell is the A type, A antigens, and also there, also that the A antigens, and also then literally that in the plasma their antibodies that is the anti-B antibodies, and also there we also that if B type that is also there in the red blood cell membrane, antigens is B, and also that in the plasma the antibody is anti-A, and we have known that there is the ABO, and also that this is the A and the B antigens on the red blood cell, and also in the blood without A and without anti-A and without anti-A or anti-B in the plasma, and they are all type. There it means that in the on the red blood cell membrane, yeah. Can you see my PPT?
Yes, 老师. Yes, 老师.
Okay. There we can see here that there it means that on the red blood cell there without the A antigen and the B antigen, and also there in the blood in the plasma naturally produce that the anti-A and the anti-B the antibodies. So that we call according to that the antibodies. And also that the antigens or other antibodies that distribution, we have now that we can also according that identify the blood ABO blood group. Yeah. So here that we can see here that the majorly that the principle of the ABO blood group identification experiment. There we just about that this experiment is that antigen and antibody reactions. The antigen and antibody reactions that we can see here that there, yeah. There we have know that there we just about that in the ABO blood group system, antigens on the red blood cell membrane are divided into majorly 2 antigens, A antigen and B antigen, and while that for the antibodies are divided into anti-A and anti-B antibodies, and also that the antigen A antigens plus the anti-A antibody or B antigens plus the anti-A anti-B antibody will cause agglutination. So we talked about that only the same type of the antigens and the same type of the antibodies then can bonding and can lead to the red blood cell agglutination. so here that based on these reactions, then can detach the whether there is A and B antigens on the red blood cell, and then based on the standard antibodies, and also then determine the red blood cell. So that the antibodies bridge between the adjustment red blood cells, the surface epitopes, and form granular collects visible to the naked eyes. See, like we can see here that if that is gutylation, and then need to more the blood cells glycation, and then because based on the antibodies bridge, and we can see here that this imagine that is not the red blood cell bonding, because that the antibodies bonding the red blood cell, and then we can see by the eyes then like that the red blood cell aggregation. This aggregation based on this bridge, this antibodies bridge. Yeah. Understand here?
Yes, Lucia.
Yeah. So here that we have now that then according to the principle of the ABO blood group identification, I hope you know that this nature is that the antigen antibody reaction. This nature of the antigen and antibody reaction. Yeah, we can see here that we just talk about that. This is also that the antigens in the red blood cell. These antigens in the red blood cell, and they are antibodies can bonding. If the antibody can bonding the same type of that, if anti-A antibody can bonding the A antigens, and then also we can see this antibody can bonding or not of the red blood cells, and then their aggregation, and their nature that is not the red blood cell aggregation, because that based on the antibodies bridge, and then due to the red blood cell aggregation. Yeah, we can see also like that is also that. Yeah.
Yes, Lucia.
Can you see that? Because the voice is stopped. Yeah.
Yes, Lucia. I can see that. Yes, Lucia.
This also that we just about that the only the same type of antigens and antibodies can bonding together, and then need to their aggregation. And also then we can see that there are if in this department that we can give that the standard anti-A antibody and the anti-B antibody, and also that in a glass we can see here that there are also that like that we can see this red blood cell 1, 2, 3, 4. There are many red blood cells, but that we can see here that the red blood cell, because basically the antibodies bridge, and then lead to the red blood cells aggregation. The aggregation, then we can see there is very small, and then very large. We can see by the eyes. So here that we call that this, and then we can see if then this anti-B antibody, it means that. Can you tell me if that's in here? Then this anti-A antibody, there is no aggregation, and then anti-B antibody, we can see that the blood, if that the blood drop in the anti-B antibody plasma the cesium, and then we also found that aggregation happened. And then can you tell me this first class? You can tell me that which type of the red blood cell? ABO red blood cell. Which type of ABO blood group? Who can tell me? From this first class, you can tell me that which type of ABO blood group according to the result. Yeah. Who can tell me? Hmm? Think again. We just about that. Then we can see here that there this NB cesium and NB antibody cesium and also this NA antibody cesium, and there is also that both the A and both NA and both NB antibody cesium, and then we can see according to this result we can see. This also there's agglutination. This is no agglutination. This is also agglutination. Then can you tell me which type of the antigens of the red blood cell? Who can tell me which type of the antigens of the red blood cell? Yeah. Hmm? Who can tell me? First, we can see here that this anti-B antibody and anti-A antibody, and also this both A and both NA and both NB. And this we can see this. And can you tell me that there is that there in this red blood cell, which type of the antigens in the red blood cell? Yeah. Which type of the red blood cell antigens? Who can tell me? Which type of the which type of the antigens of the red blood cell? The first, this first, yeah. We just talk about that. There is this red blood cell in the membrane is A is that membrane is ABO blood the first. Yeah, thank you. Thank you, Lisa. Yeah. Oh, O type. O type, it means that there is no A antigens and or B antigens in the red blood cell membrane. So that is we have known that this is the O type blood group. And then I have know that in first that we know that the red blood ABO blood group the antigens and the antibodies and then can classify the 4 types of red blood cells, red blood group. And also then the red blood group, we know that which type of the red blood group and also which type of the antigens and antibody in the red blood group. And then only that we have you need to also that we also know that the organization it means that the antigens and antigen antibodies reactions. And these reactions, we know that by these reactions we can identify the antibody antigens on the red blood cells, which type of antigens, and then we can identify the product. Yeah, very good. And then we will do the experiment for that. The Eugene and also the materials that for that in we should have the standard NTA and NB and special. The first that and also to speak and also have the glass side. Their glass side and also then first that we should drop one drop of the NTA and NB on the glass side. And the second that we should have the needles and then need to the blood in and then need to and also drop one drop of the blood into the NTA or NB antigens, and then we can confuse that, and also then we can see, we can observe the reactions. Is that we have now that put one drop of the NTA and NB cesium on the both sides of the glass slide, and also mark the word NTA and NB. And there is also the glass slide, and they all we normally that we do drop one drop NTA and one drop NB, and then we can see here that drop A and drop B, and drop A and drop B. And also there, also that we then punch there. Also that the defines the end of the ring finger. We can see here the ring finger of the left 75% alcohol cotton. And also there, we usually use this the ring finger. Then because they are the ring finger, and they need to the small damage. And also there to be also they puncture the skin with deposit blood. This needle we can see here that this needle, and also then we also that this needle, and also that we need we also need more blood, and then you and also we use our toothpick to drop one drop to the blood into the NTA. The mix together, blood into the NTA cesium and mix together, and then also that use another toothpick, and then also drop to the drop of blood to the NB cesium and also mix together. Mix together is means that need to the antibody and the antigens touch the folly, and then can easily to reactions and bonding. And also that mix well, and also that after standing at room temperature, and then we take that this in the table, and also they observe there whether there is occultation. There are about that observe about 4 and 10 minutes, and then we can see that this is the blood and the standard cesium can have the occultation. Yeah, here understand.
Disclosure.
Yeah. And also that we just about the picture that and also there we should also that the cottons and then the blood stop bleeding stop still stop the bleeding. And the first that we should that this feature that with 75% alcohol the alcohol cotton ball. Yeah. And there we mix that we can see here, observe there we can see mix where and then we can see observe the result, and then we can see here observe where there is a granulation, and here that we can see the result. The result we can see here that like that there also that this anti-A antibody and anti-B antibody, and they are we blood the same the people's blood, and also that the blood in this anti-A and this anti-B and mixed together, and also that we observed last for 5 and 10 minutes, and then we can see the result. Then result, can you tell me that which type of that we this is A is we can see here that is also that is a large that is also we call that granulation, this A, and this B and B is not a granulation. Then we have now that because this granulation is means that in this red blood cell have the A and antibodies antigens, and also that in this red blood cell there is no B antigens. It means that this blood group, ABL blood group, is A type group. Yeah.
Yes,
Understand
Lasha.
here? Yeah,
Understand,
understand.
Lasha.
And also there we can see here that this is like the night cloud is not the granulation. And there we can see a little this that we can see here that this granulation. Yeah. And also that this result, I hope you know that. And then we can see also that is also there we can see that immediately that is all bounding, the bounding. Yeah. They also that we can see that this is also the anti-A antibody and also the A group, and then we can see here that is also that the bounding, and also the anti-B is also they cannot bounding. Yeah. Yeah, we can see here that like that also that we can see that this A group antibody system. There we can see like that this A, and this means that if the anti-A antibody oxidation phenomena is type A and type A, and then we can see here that this anti-A and both NB, both NA antibody and both B antibody are oxidation. It means that this blood is this red blood cell have the A antigens and B antigens, and then is AB blood group, AB type, AB blood group. And we can see here that there is only anti-B oxidation. It means that this is a B group B. We can see here that the A and B and B and NA all know that oxidation. It means that the blood the O type of the O type or go O. Yeah. Can you understand? Do you understand here?
Understand, Lasha.
Yeah. This phenomena also easy to observe, and then this result, I hope you know that, and this immediately that the first result, and there is also the second result, also that then if we see that this result, and then we know that this is the we can identify the which type of the ABL blood group. Yeah. So here that this impairment that I hope you understand that and discussing. I hope you know that what is discussing. According to the impairment, discussing is according to the our impairment result. The result and the result is the phenomena. We can see here this is the result. Result is the phenomena. First that we should describe the phenomena. First, describe like that we can see we describe that like that the result we describe that this describe that the result that NTA is oxidation, NB is not oxidation, and then we can know that this A type group this result. This result the picture. This result the word. We should know that result include both the pictures and both the word. We should describe the result. And also the describe result and there we should this also like the describe the result. This picture and also the result. And this result we should analysis result. It means that we should analysis that why this phenomena happened. And then this based on the knowledge we have studied the physiology. So that discuss it means that analysis the result. Yeah, understand here?
Understand, Lausha.
Yeah. And this experiment 4, I hope you discussing there for that the distribution of the antigens and antibodies for the ABO blood group. This that only we master this the distribution and then we can know how to do and identify the ABO blood group. Yeah, this is the knowledge or the basic knowledge of the ABO blood group, and also that the second that the antigen oxidation it means that the antigen and antibody reactions. Also I have know that, and there is also that the cross matching. The cross matching. What is the cross matching? I hope you understand also that what are the principles of the blood transfusion. Yeah. This AB blood group, do you understand?
Understand, Lausha.
Okay. And also I hope you
Yes,
read the experiment report. Yeah. The blood cell is also very easily to understand, and also they are often used in the clinical work. And also, I hope you master that distribution of the antigens and antibodies of the ABO blood group. And we master this knowledge, we easily to understand and have to do that. And because that we know that before the blood transfusion, you should ask the patient to do 1 ABO blood group identification, 2 and cross matching. Yeah. This is the experiment 4 we study here, and we have a yes. Okay.
Okay, Lausha, thank you.
Have a yes. 10 minutes later, we are continue the experiment 4, experiment 5. Yeah. back. Golden boy, can you hear me?
Yes, social, I can hear you.
Okay. Can you see my PPT?
Yes, social.
Okay. We will see that the payment of this auricularly injection method. Also, that we have the dependent one. We have described that how to do the also here neck operation or surgery, the neck operation, and also there's some the neurovascular separation, and also there majorly that there is the intubation, tracheal intubation, and also that the record the rabbit's the breathing curve, or we also that respiratory curve. And here that there we do you observe that after this surgery and do the step observing the movement that the side and also the hydrogen increase, and then lead to the drug effect on the respiratory movement, and also that there are some the nervous that movement, you know that. Okay, and there first that we before we study that the impairment respiratory movement, I hope you know review the knowledge that what is the respiration. The respiration, we have know that between the bodies and the external environment, the gas change. The gas change means that inhale oxygen, and exhale the carbon dioxide. This that it means the gas change. Between the bodies and the external environment. And here that the process, the major process of the respiration include that the military contact one, the internal the transport of the gas in the blood, and gas transport in the blood. Three, also the internal respiration. There are this majorly the three process of the respiration. Yeah, do you remember? Do you remember that the three basic process of the respiration? Yeah. Yeah. In the gas transport, respiration, extra respiration, two types also that the pulmonary ventilation and the gas exchange in the lungs, and also that the pulmonary ventilation. We have know that what is the pulmonary ventilation? Do you remember? What is the pulmonary ventilation? Who can tell me? Who can tell me what is the ventilation? Yeah. Process of gas exchange between the lungs and the external environment. Yeah, thank you, Masali. Yeah, very good. It also means that the gas change, the process of the gas change between the lungs and the external environment is means we can see here. The lung is also that is also in here, that is the elbow. The between the lungs and the external environment, there are gas change here. And then who can tell me that what need to that this gas change means that the gas movement? Yeah, the gas movement we have know that the gas movement needs the force. Yeah. Then what's the directly force needed to the gas movement? Do you know? The permanent ventilation is means that the process of the lung between the gas, process of the gas change between the lungs and the external environment, and then this is means also that gas is change the gas movement. The gas movement we have known that needed to force, and then who can tell me that the directly force of the gas change? What is the directly force of the gas change? Yeah, who know that? Who know that? The gas movement. What is the directly force? What is the directly force needed to the gas movement? It means that inhale or exhale is also that the gas out to the body or into the body, and then the gas movement. And what is the directly force? Yeah, who knows that? Hmm? Who knows that the gas? Why the gas can body or into after the lung or into the lung? Who can tell me? Direct force. Yeah. Who knows that? Who can tell me? Can you tell me? Yeah, 李一通. Do you know that what needed to the gas after the body or into the body? Back to that.
In body.
What?
In body.
I can't hear clearly.
Into the body.
The gas moving after all the gas is done. So that basically that outline that is comes after the body outline move. Do you understand here? Yeah. The period, know that. We know that then when the period of the oxygen in the lung is lower than different period. Yeah, inside the body, very good. The period, different period. Thank you, 林姐. The period is different than the external environment. So here that the carbon dioxide of the body. At the same time, the oxygen period in the external environment is higher than the lung or the direct period. Yeah, very good. And also that we have know that the different the external environment the lung cannot respond by itself. So here that the lung change because that the lung visually that the mass or the muscles part the muscles see here that the thyrox expand or thyrox expand and then to the lung expand. Lung expand and then to that the lungs the interpulmonary the period decrease and then the atmosphere period and then we can see here that the gas or the oxygen into the lung. At the same time, if the respiratory muscles relaxation and also they need to the thyrox reduction and also need to the lungs reduction and also they need to that the period is also that the period is the interpulmonary period is higher than the higher than atmosphere period and also they need to the gas after the body. So that expiration. Yeah. So here that the primary force for the primary force of the primary muscles movement, and they need to the primary ventilation. The direct force is that the period. The primary force is that the respiratory muscles contract. And they need to that the gas the pulmonary ventilation. Yeah, is that here? Yeah, and there so here that there we know that there we found the principles that first that we talked about of the respiratory muscles controlled by the respiratory center, which moved the gas in then the blood, if this is the oxygen and the carbon dioxide, and also the constitution of the hydrogen, and then also the regulate respiratory movement and some respiratory central, and then also that affect the respiratory movement. And also some that the pulmonary stretch reflex, imagine the vigorous nerve. There we don't do that. Okay, and here that the pulmonary materials that first that we do this animals that is rabbit, and there are the experiment like that the animal. We will do the animal the surgical equipment like that the scalpel and surgical signals and also the homeostatic phosphates and glass minute needles and also that arterial clip and silk sleeve thread, and also some the also the imaginary here that is tracheal intubation, and also weight, and also that is respiratory transductancy, respiratory transductancy, and also some the microcomputer singular recording system. And the medicines imaginary that we use that we will shoot the surgeries, so that we should take the you should take the username and 20% username, and also some that 2% the lactic acid, and some the drugs like that the nicotine, also that 100 I'm sorry 100 there is also 100 the 100 the Nixon. Yeah. In instrument the collections and the calibration, and the second that we should waste the rabbit, and also that the should fix and showering, and also there you deploy the neck, and then do the neck surgery, and also that we should do the tracheal intubation, and also that the observation. First, that we should that take the rabbit and weight, and or let's see the imaginary that 20% using and by the 5 minutes 5 microliter the kilogram, and also fixation, and also that the back position cause the fixation, and the surgery that we surgery like that also the surgery like the neck of the skin was cut along the midline, and also that the fat skin was cut, and also the muscles were buttoned separate, and then the tracheal is pleural. The tracheal pleural or single cotton thread was put through for use, and also under the testing chest, and also clap the rabbit trim on the side tracheal intubation with the clip, and also that adjust that and also open the microcomputers operation, and then we should record the respiratory curve. Yeah. And here that we first that we just that the weight the rabbit, and also that analysis. Then we can see here that we use the ear vein injection. Also that we also that we should observe the rabbit respiration movement, and also that his the pain reflex, and also that we should his eyes. Also that we can see that if the muscle also the muscles contraction, if the muscles relaxation, and also that the respiration movement is slowly and stable, and also that the pain reflex disappear, and then this means that the rabbit in a stable analysis situation. Yeah, and also then we should fix the rabbit in the rabbit table. With the rabbit table, we can say that fix the rabbit table. Yeah, and also that we should cut the hair. The neck of the hair, we cut also we cut the hair, and then skin of the neck, the skin of the neck, skin and the neck of skin, and the little skin, and then we cut the skin, the cut the skin along the middle line. We can cut the skin on the middle line, and then we first that we cut the skin, and then we first separate the softer tissue, and also that we can see that the muscles, and first the softer tissue, and also then we employ the tracheal. We can see here that employ the tracheal. Yeah. And also there are you put track and also we majorly that distinguish that the villus lobe and there under the lobe that villus lobe there is two sync threads for the each of them. Here that I hope you that you also found that the villus lobe and also you found the article arteries and also there. Also the vein, the neck veins, you shouldn't know that. This the impairment you should first that you know what is the trachea, and the trachea that we can see here that should that was the separated, and also that the cotton thread was threatened under the also under the tracheas there we should take a cotton thread. And also that we should cut under that the bone. We can see for the about the third and above the fourth, we cut the trachea and the T-shaped what is the T-shaped in incision. That we first the T-shaped inverse T-shaped. Yeah, and also we can see here like that. We cut the trachea. We cut the trachea, and also that we should insert in make a insert inverted T-shaped incision. There below the carrot cartilage, and there we cut that. We can see here that they cut the inverted T-shape incision. Yeah.
Oh, sure.
Yes, here. We have like that. This is the tracker. We force that the inverted T shape. It means that the T shape, the T shape, inverted T shape. And then we do this incision, and then we should intracerebral intuition into insert this the incision. Yeah. And also that we can see here that this is also that tubulus that collect with the respiratory transducer, and then collect to the computer or the the computer. Yeah. Also we can see here that there we should do a inverted T shape. And there here we can see under that there is first the T shape. Can see here that. Yeah, and also then we fix the intuition in the trachea, intuition in the trachea, into the trachea. And also there we should thread and then fix the cotton thread and fix the intuition in the trachea. Yeah, can see here that. Here at the end here. The cotton thread, not in the thin thread. And there, if a thin thread needing tracker damage, and the cotton thread cannot damage the tracker. And also that that means that the air can into the intuition and then into the airway and into the tracker and then into the lung. Don't by this the intuition collect the respiratory transfer can record the respiratory curve. And also that we can see here that this is also that the nervous vagus loop, and also the vagus loop, and also that separate that show that the vagus loop. You know that, okay. Yeah, then I have know that this we do the inverted T-shape, the incision like that, the tracker incision, and also that we should insert the intuition and into this incision. Yeah, and also that the separate the nerve, you know that, okay, separate the nerve don't. And then we just about that we can see here that this collect the respiratory transduction which we can record the respiratory curve. We can breathing curve or respiratory curve. And then we can see here that this is the curve like that. We can see here this is the inhale inspiration, and also this is the actual expiration. Is that we can see here that the breathing curve be made up that the inhale inspiration and the expiration curve. Yeah, present. And then we can see here that this is that it means that the rate of the breathing curve, and also that this is means that the aptitude of the Just here.
Yes, Lauscher.
Yeah.
Yes,
This curve,
Lauscher.
I hope you know that this is means that inspiration. This means that expiration is also the inhale or exhale. Is inspiration, expiration, inspiration, expiration. We can see here that, and there is also the high, the height. It means that is the aptitude of the inspiration curve, the aptitude, and also that this means that the rhythm or the rate of respiration. Yeah. And then we should observe the influence factors of the respiration movement. First, that the hypsio. We should use this synonym bottle. Synonym beta is this in here. This is synonym. Synonym he can lead to that he can absorb the carbon dioxide, but that there we can see this collect the intuition, and then we can see here like that this rabbit we can see here that this collect the air, the gas, the air atmosphere. This collect the computer, and then we take the bottle in here, and then the rabbit can only inhale inspiration the bottle of the air, and inhale there we can see here that they are inspiration, and then lead to the carbon oxygen decrease, but that the carbon dioxide doesn't increase, and there we can see here that lead to the Hapsio model. Can I send? Yeah. The bottle is that it have the pseudonym side because the then due to that he inhaled the oxygen, and then he exhaled the carbon dioxide. The carbon dioxide can be absorbed by the soda. Yeah, answer here.
Understand, Lauscher.
Yeah, and then this republic that the Hapsio model, and the second that and also then we can you observe the curve change, and then if the curve change happened, we stop this schedule. And the second that also due to the curve change, the curve recovered, and then we do the next. Let's increase the dead space. In the dead space, we use a long rubber tube. A long rubber tube also that we collect that. We collect this. We have a long rubber tube. We collect here, and then means that we not that need to that the increase the dead space, and also we also observe the curve change, and the curve change increase happened. Also we stop that. And third, that we also the injection, the ERVM, the injection that the also that the 3% lactic acid, and so then we also observe the curve of the curve, and the observe also that recovered also that do that their the nicotine we have now that he is started the respiratory century, and also that we also can see the curve change, and also they need to so change so the change recovered we do that the incomplete that curve that we the half the we tightly, and then we see the curve change, and also the, to the rabbit die, and also we can curve change. So here that we can see that curve changing like that. The experiment result. We also found that the Hubfield forced that the curve increase, and also that the amplitude increase, also the rhythm increase, and also there we stop also recovered. Also that we also increase the dead space, also that he is increase. Also that we also that this increase we can see here that also that recovered also that we had do that the 3% the lactic acid, and also we can see here this is the slowly increase, and also that slowly increase also that we immediately we and also then incomplete or the rhythm is decreased, the amplitude is increased, and that the rhythm or rate is decreased, and also that completely that we can see here that increase, and also even disappear. The curve disappear. It means that the rabbit die. Yeah, this is the experiment result. Can I understand?
Understand,
Yeah.
Lusha.
And this the experiment result then according to the experiment result, you first like the Hubfield, and also then we have studied the knowledge that the physiology we know that why the Hubfield the respiratory movement increase, and then I hope you know the respiratory movement. There I hope you know that how to
Understand, Lusha.
do you understand here?
Yes, Lusha.
Yeah. And also that the dead space, the increase the dead space, then the why we can see here why the movement increase. There are also that I hope you know that blood the hydrogen concentration increase, and also then can regulate the or need to regulate the part movement. Yeah, the nicotine limiter is means that it is the central is part of central excitation drug, and then he need to the expiration. And also, here that this I hope you know that how to according to the result. I hope you know how to discuss why this result happened or this phenomenon happened. Yeah.
Okay, Lusha.
Okay. That discussing. I hope you know that the experiment result that I hope you that the this write the curve. Okay, write the curve like that. The first that Hubfield before that, and the also the rate range the aptitude also the rate. the table, the result, and they are, and also that the and also that discussing. Please for that, please track the change in the heart rate and the aptitude for Hapsia inhalation and enlarge of the dead space and the injection the lactic acid. That the cause change in the respiratory curve. Yeah.
Yes, 老师。
Okay. This class we will talk about here. I hope you now have that review the knowledge of the respiratory system, respiratory system, or respiratory system, respiratory functions. Yeah, the chapter 5. I hope you review the knowledge and then discuss this. Discuss it, or it means that the collect the knowledge and the experiment. I hope you know that how to collect, how to keep the relation between the knowledge and the experiment phenomena. Yeah. Okay, this class we will talk about here. Okay.
OK， 老师 ，Thank you， 老师。
Okay, goodbye.
老师。
OK， 老师。
we can any questions we can discuss the dining group. Okay.
好的，老师。老师，
OK，
Thank you。
谢谢您，老师，再见。
再见。
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 谢谢老师，再见。